Today, we are diving into a truly fascinating new study that has uncovered what you might call a secret failsafe, a hidden backup system tucked away inside our own genetic inheritance. It's an incredible story, part biological mystery, part brilliant experiment, and it ends with a discovery that could really change how we think about heredity. So let's kick things off with a pretty huge question. Can the things your ancestors went through, what they ate, the stresses they faced, can that actually leave a mark on your genes today? And I'm not talking about changing your DNA sequence. We're talking about something way more subtle. The field that's looking into all this is called epigenetics. And here's a great way to think about it. Imagine your DNA is this incredibly long sentence. Well, epigenetics is like the punctuation, the commas, the periods, the exclamation points that tells your genes when they should be read and when they should be ignored. One of the most common types of this punctuation is a tiny chemical tag, a process we call DNA methylation. It doesn't change the words in the sentence, but it can turn a gene on or off, which, as you can imagine, completely changes the meaning. Right, so for years, scientists have been locked in this debate. Can these little epigenetic marks actually be passed down from a parent to their child? It's a massive question, but proving it one way or the other has been, well, notoriously difficult. I mean, how can you be sure that something you see in a child is because of an inherited epigenetic mark and not something in their diet or their environment or 1,000 other things? And that is exactly where our story really kicks into gear. To finally solve this puzzle, a group of scientists decided they needed to take a much more direct approach. Which brings us to our first section, editing the epigenetic code. So instead of just sitting back and observing what happens in nature, these researchers over at Japan's Gunma University decided to intervene. They basically asked, what if we stopped guessing? What if we could go in and directly edit that epigenetic punctuation right in the sperm and egg cells, the cells that actually pass information to the next generation, and then just see what happens? To pull this off, they built this incredible tool, a molecular eraser, if you will, with 3 key parts. First, they took a disabled version of the gene editing tool, CRISPR. So instead of cutting DNA, it acted more like a GPS, guiding them to one very specific spot on the genome. Second, they attached an enzyme that was the eraser itself. Its whole job was to remove those DNA methylation marks. And finally, they aimed this whole contraption at one single target, a gene called H19 in mouse sperm, a gene they already knew was absolutely critical for growth. And would you look at that, it worked perfectly. What you're seeing here is the proof. In the top image, you have normal sperm, and you can see the H19 gene is just covered in those little black dots, which represent the methylation. But now, look at the bottom panel. This is the sperm from their edited mice. It's almost completely wiped clean. Those black dots are gone. So the key takeaway here is they now had sperm with a very specific man-made epigenetic change. So the multi-million-dollar question is, what happened when these mice had pups? Well, this is where things get really, really interesting. So let's move on to section 2, a message partially inherited. The very first result was, well, it was striking. The pups born from the fathers with that edited sperm, which you see on the left, were clearly noticeably smaller than the normal pups on the right. This was definitive proof that the epigenetic change they made in the father's sperm had a direct physical effect on his children, causing a type of growth problem that's actually pretty similar to a human condition called Silver-Russell syndrome. And we're not talking about a small difference either. This chart really drives the point home. You can see a significant drop in the birth weight for the pups whose fathers carried that molecular eraser tool. The edit had real consequences, proving that a mark in the sperm could definitely be inherited and could affect the next generation. But just when they thought they had it all figured out, the story took a wild twist. When the scientists looked at the DNA of these smaller pups, they fully expected to find that H19 gene totally clean of methylation, just like it was in the dad's sperm. But they didn't. Incredibly, the epigenetic mark they had so carefully erased had somehow been partially written back in. This, of course, kicked off a brand new mystery. The message was gone in the sperm, but it was reappearing in the embryo. How in the world is that possible? It was like the embryo had a backup copy of the information stashed away somewhere, a ghost of the original epigenetic mark. So naturally, the hunt was on to find this mysterious backup system. And that brings us to section 3, searching for a backup. So the scientists basically became detectives, tracking exactly when this recovery was happening. Let's just follow the timeline here. In the sperm, 100% erased. In the zygote, that's the very first cell right after fertilization, it's still erased. But then, a few days later, in the very early embryo at what's called the morula stage, you can see it starts to come back. And by the time the pups are born, it's been significantly restored. So the key clue was that something was actively rebuilding this mark in the first few days of development. All the clues started pointing toward a different kind of epigenetic mark. This one wasn't on the DNA itself, but on the proteins that DNA wraps around. They're called histones. You can think of them as the spools for our DNA thread. And they zeroed in on a prime suspect, a specific mark on these spools called H3K9me3. The new theory was that even when the methylation was erased from the DNA thread, this molecular bookmark on the spool was left behind, telling the embryo's machinery exactly where to put the methylation back. Okay, so they had a suspect. They had a really good theory, but in science, you have to prove it. And that brings us to Section 4, the epigenetic recovery system, and the final brilliant experiment that really cracked the case wide open. To get their smoking gun, they designed one last experiment. The logic was simple. If H3K9me3 really was the backup template, what would happen if they removed it too? So using another targeted editor, they went into the embryos made from the edited sperm and specifically erased that H3K9me3 bookmark from the H19 gene. It was like removing the suspect from the scene of the crime. And the result? It was absolutely definitive. Just look at this. In the control embryos, where they left H3K9me3 alone, the DNA methylation was partially recovered, exactly like they'd seen before. But in the embryos where they erased H3K9me3, the recovery failed. Completely. The DNA stayed clean. They had found the backup system, and they had just broken it. And taking out this backup system had serious consequences. The embryos where the recovery system was disabled, you can see them on the bottom, had even more severe growth problems than the ones that only had the first erasure. This proved just how biologically critical this hidden recovery mechanism really is. So let's take a step back. What does all of this mean for the big picture? Let's get to our final section and see how this discovery is, in a very real way, rewriting the rules of heredity. Okay, so here's the final verdict. We now know that this hidden backup system for our epigenetic memory is real, and we know that a histone mark is the key. But here's a crucial detail. While the father's edit affected his direct children, that's what we call intergenerational inheritance, the backup system actually fixed the error before it could get passed on to the grandkids. So it was not a transgenerational effect. The system essentially broke the chain of inheritance after one generation. But like all great science, this amazing discovery opens up even more questions. As the scientists themselves say in their paper, it doesn't seem like H3K9me3 is being transmitted directly from the sperm. Something else is telling the embryo to place that bookmark there in the first place. The ultimate source of this inherited memory is, for now, still a mystery. And that really leaves us with a final, fascinating question to chew on. This study uncovered one absolutely incredible biological failsafe. So you have to wonder, what other hidden backup systems are out there, quietly shaping our inheritance in ways we're only just beginning to imagine?